I'm going to start off with one question, and then we can take questions from the audience as they come. One question I have for both of you is really like around data. On like, you know, the side of Odyssey and Patrick is all about like making the data come together as large as possible, standardization, because knowledge may be even hard, and so doing it in a way where you have like background information from the data can help you. On the other side, like data certainly helps and plays like a giant role. Maybe you told me like a slightly different story where like here's a success where we didn't have to go push as far on the data side. And so like where do you see the future in the organization of data and in sort of the way machine learning view of this? Like are we going to have like representations that are shared across health records as part of Odyssey in the future? Cannot hear. Too low. How's that? Better?
Keep talking.
Better? Any better? Can people hear me? No?
Hello? Can you hear me?
Yeah, that sounds, I think our mics are on. His isn't.
Yeah.
You're on a little bit.
Now can you hear me? Is that better? All right,
Yeah,
good. Thank you. Thank you. Well, I said a bunch of stuff, so I'll say it all again. So my real question, just summarized, is like, you know, what's the future of how we're going to keep track of data? You know, are we going to organize them together into larger and larger bits? Will we also organize like, you know, pre-trained machine learning bits around it, like representations inside of Odyssey is what I said. Maybe there are similar questions around genomics, but like, what does the world look like a few years from now in terms of like how we organize the data, how we organize large models trained on that data, and what we share about it?
Do you want to go? I can go first, yeah. So honestly, a lot of this, what I just showed you, has been possible due to large funded consortia that we've been part of for more than a decade. So since the Human Genome Project ended in 2003, NIH funded many large open projects. And one of the coolest things about those open consortia has been a lack of data embargo. So the thing that we really worked hard on is to make sure that we got experimental groups on board who are willing to openly publish our data as it was generated, right? So we didn't wait 10 years or 4 years or 3 years before the papers were published or before whatever high-value targets were achieved. The data was released pretty much on a streaming fashion. And it has really, in many ways, powered a lot of these revolutions in machine learning that we're seeing. So at least in the genomics front, actually, there is a ton of completely open access data. And there's been a huge effort over decades to curate it, QC it, make it machine learnable. A lot of the models we now have are hosted in model zoos. So there's been a lot of organizational work on building the infrastructure and the capacity to enable these kinds of models.
Great. So I'll point out the type of research we're doing is often rare effects of drugs. And by the time you pick the drug and the side effect and the population, you need like 1 billion or hundreds of millions, a population of that order of magnitude in order to measure the effect size for rare side effects. So we're stuck needing this, but in order to, the US only has 5% of the world's population. We can't do it in the US alone. But we have strict privacy laws or varying privacy laws across countries. So we created a federated database. So it's true, that's a conservative estimate, is that 928 million unique patients have been converted to our data model. A subset of that actually participates in studies. It's a voluntary network. We can maybe get to half that. So we're like getting close to half a billion. But you have to be able to do your research on a federated network. That is, to run out on each of these databases and then combine the information in a way that there's no leak of privacy. So that's an important part for that to move forward. One thing that has made it easier is that we've only been using clinical data so far, so like administrative claims and electronic health record data. Now the US All of Us research program is using our OMOP common data model, and there they are combining genomic and clinical data. That's on the order approaching 1 million patients in that database, which is similar to, say, the UK Biobank and other will be approaching that soon. Earlier in the day, I was hearing and overhearing conversations about, well, I got to Mimic and I got one other data set, but I need a lot more to do anything. And then Patrick goes on the screen and says, well, I have 928 million people. Do you want it? And so let me just take this opportunity to say, how do you get it? And so it is a lot of work. It's a voluntary network. You have to convince people to run your study, basically. So it's not funded by anything in particular. It's just this group of people working together. And so generally what you do, so people who from this community, like Nigam Shah's group or David Sontag's group, you pick someone, a postdoc often in the lab, who throws themselves into Odyssey for a year, gets to know everybody, helps other people, and in return they help them. And then when you come along and say, oh, I want you to run my study, it's going to cost you a month of CPU time on your server. Do you mind doing that? And they'll say yes to that because you've been helping each other. So you have to really throw yourself into Odyssey, join the community, be part of it, but then you get access to something that is otherwise unheard of.
Great. I did mention when I was muted that I'll take questions from the audience as well too, so it's not just me talking. But I do have a follow-up for myself. Is like, what about the other bits of data that are popular among the posters and papers that you see here? You see a lot of waveforms, images. You mentioned wearables very briefly. Is there a future for that to be incorporated in a way that is easily accessible, either in a common model or a federated model?
The All of Us research program is pulling in survey results, getting some mobile monitoring stuff in. So they're probably the head of the pack for working with the OMOP common data model. And then there are various consortia that are trying to move forward, and some cases people trying to make some public data sets in individual areas. But as a big comprehensive thing, I think for me, I don't know, unless people know of other ones, the All of Us research program is the one the closest to getting the biggest heterogeneous set. There may be a question.
Good.
Can you hear me? Okay. Thank you for very different talks on very different topics. So I'm trying, if okay with you, to ask question for each of the panelists. So maybe with the first talk, and I'm not going to put the guest panelist on the spot because it was a separate presentation by a different person. But if you recall on Patrick's presentation, there was a slide that he was emphasizing that where we are, and in terms of machine learning for healthcare community, bespoke models, and you define a problem, you work on it on weeks, maybe years, to ultimate place to be seconds, that you instantly look at the evidence and you are able to basically have an evidence-based approach, right? So, I mean, for a second, imagine that over a second we can achieve that ultimate goal, that golden state, right? So how then would be the role of this community, machine learning for healthcare community, right? Wouldn't that be in that scenario that everything is standardized, you have existing techniques as standardized data, what would be the role of the technical community, machine learning for health community in that sense?
Okay, so first, now I may have misread it, but I thought that slide of Patrick's was more referring to population level estimation and not predictive models. And when he says it takes a year to do a study, he meant the FDA publishes on their website a question, does a drug cause angioedema? And then people go out and run the study over the course of the year, and finally we get the answer much later. And what he wants to do is run those studies ahead of time, do everything. So we'll put it back up on the internet soon, but howoften.org is what's the rate of every side effect after every drug. Non-causal for the first version, so it becomes feasible, but it's still useful. If the rate's very low, then it doesn't matter if it's causal. So that's an example we're doing. Every drug, every side effect. That rectangle that Patrick showed in the beginning with the red dots would be a full red rectangle full of evidence. So that's an example of doing something ahead of time. So the seconds is because all you have to do is go in the user interface and look up the answer. But we can't guess everything we're going to need to know. So forgetting prediction, I'm going to get to that in a second, even on this population level estimation, we're going to do one class of questions at a time. So like even the epidemiologists still have a job. They're going to be ones pre-generating all these things, and we're going to have to march through medicine, but we're never going to get through medicine. It's an endless series of questions that we have. So like I don't have any worries about a role for epidemiologists in this first half doing that. Then we have putting this into practice and like helping the doctor at the bedside, and that's more the predictive models that for this particular patient. And that's, I think of that as more this community. And I think, you know, we're the engineers who are building these systems and maintaining them and improving them. And, you know, this is really hard. So I'm not worried about the next 100 years of being done with it. So I think that we remain the engineers developing the methods and running this. And then he's just saying the evidence that we generate or the systems that we build become open source, and then people can get to them quickly, but we'll still be doing the next ones.
Right. Okay. Thank you. And I mean, very quick question for the second talk as well. And I'm not trying to pretend that I was able to understand everything. I have to.
I hope it was not completely all that.
Right. I admit that, like, to the point that I was able to follow all the presentation, there was a part that was quite interesting. And I think in your language, you were also calling it like noise versus like the more interesting spikes. So, and then you later showed that like you can join these two parts, and one of them is regressing out the effect of the other one. So my question was that like how can you explain? I mean, if you label it or call it noise, what is the explanation for being able to identify those patterns? Like why there should be a model that can nicely capture those so that you can filter them out, right?
Yeah, very good question. So most of the experiments, these molecular experiments that are done, they're using clever biochemistry, right? So the examples I showed you today, the experiments that we're looking at, as I mentioned before, they're using specific enzymes. And these enzymes, the goal is to identify regions in the DNA that proteins are next to, and these enzymes will preferentially nick the DNA right there, right? But these enzymes are enzymes. They have their own sequence preferences. And if the goal of generating this data is to understand sequence features driving the biology that the experiment is trying to capture, unfortunately, the experiment is going to capture not just that causal underlying biology, which is driven by sequence, but just due to the technical artifact of using a specific enzyme with a specific sequence preference, that signal gets injected into the experiment as well. So just to put in context the novelty of this work, when I present it this way, it seems obvious, but most of the time people take experimental data as the ground truth. And the point I was trying to make is it is not the ground truth. It is the ground truth confounded with experimental artifacts. And so you cannot do blind machine learning on it. That was my point, is if you just train a model and you do stuff with it, you will, just as in healthcare, where if you train a model naively on data you just pull from somewhere, you're not only going to learn causal signals, but lots of confounding, spurious stuff, and then you deploy it in the wild and it's going to be mayhem. Luckily for us, there's no mayhem if you do it, but you get wrong predictions, right? But the key point is you can, in fact, if you're careful about it, figure out tricks to learn models that only learn biases and then be able to pull those biases out. So in some sense, even in the molecular world, the notion of modeling biases through machine learning is relatively new. If there's one point you take home, maybe that's the point.
Makes sense. Thank you.
Okay.
Hello. I have a question for the second talk. In particular, I'm curious about the in silico mutagenesis. First question is, I guess generating in silico, it makes me think of generative models, right? But as far as understood, is it could you give more details on how exactly you are doing the in silico
yes.
part? And then follow up, I'm wondering how does so are you thinking also of the out of distribution? So, because I could imagine that basically you want to be able to simulate what is scarce or not available in your data set. And so how are you thinking about that, potential inductive biases or limitations or possibilities of that out of distribution? Thank you.
Excellent. Both questions are very good. So the first question was regarding what is in silico mutagenesis. It's not a generative model. It is, all we're doing is we are making mutations to the input, right? But those mutations are not random. They are mutations you actually see in patient genomes or in individuals, right? So it's just, you're taking one version of the sequence that you see in some people, and then you take another version of that same sequence, usually with a single base change, right? And just push it through the model. The model makes predictions. You look at the delta from the prediction, and that gives you an effect size of that mutation. That's it, right? But you're also right that the same model can be turned on its head and be used as an oracle for synthetic DNA design or optimization or editing. And we've done some of that too. And interestingly, these models can go pretty out of distribution. So they have very interesting applications for, for gene therapy, for example. You want to activate a specific gene in a specific cell type through viral delivery. You want to design an element that will drive that gene only in that cell type at a specific time point, right? You can use these kinds of models to do those designs, and they can go pretty out of distribution, which is interesting. That being said, I think an interesting problem is to figure out the boundary of the model, where it actually makes a confident prediction that is completely wrong, right? And currently the models are not trained on genetic variation directly, but we expect that as we train them on actual genetic variation data with corresponding molecular data, they will get better and better at more and more out of distribution predictions.
Hi, I'd like to thank the panel for this great discussion. I'd just like to follow up on Rajesh's question on general data. Also, it follows up okay, there's a lot of, I guess, foundational models that a lot of people want to train. Is that possible in healthcare, and what does that even look like in healthcare? Thank you.
Okay, so which kinds of sorry, sit down already. Foundational models of what sort?
Yeah, I guess, whatever your vision of a foundational model is, I guess it changes for a lot of people, but when you think of natural data, it includes imaging, it includes video, it includes audio, it includes whatever. So just whatever your vision of it would be.
So models that you would pre-train, say, if you had a large language model applying it to the time series of an EHR, and how could you pre-train such a thing and then further train it through transfer learning kind of thing? Is that what you're referring to?
Yeah, also maybe perhaps a model I guess in bespoke models we're doing binary classification or a couple labels, but maybe something that actually represents more of a human thought process where you kind of understand the whole scope of healthcare, and then you can apply it to different things.
I mean, we do those experiments largely by picking databases of a medium-sized chunk. It can't be too small or you can't get enough information out of the database. Well, we don't have access to the 900 million patient single pooled database. So what we tend to do is build models at sites that have some large population, 100 million or 6 million or something, and then figure out how to combine the information among them. As far as there being we've always strived to build this model of the healthcare process being an example, because there's the physiologic process, but then healthcare puts these, you could put it as noise or whatever you want to call it. Sometimes it's the healthcare process we're trying to fix, so it's actually the primary model of what it does to the data, right? Because the data are, for example, you're only measured when you're sick, and that's something that should be incorporated to any model of how when we use EHR data, knowing that we're only going to get the sick data and we're not going to get data from when they're healthy. And that's something we should be able to learn from the data, right, and create this kind of large healthcare process model. So in practice, researchers that I know doing it are doing it, one large database at a time. They're not really moving it across databases. They're really just running it in each database and publishing those papers for the most part. I guess it could be moved. Usually, when we're combining data across databases, it's something much simpler, like one question. Like it may be 10,000 of them, but the primary question is this drug, that side effect, which is more. That's the kind of thing we're actually sharing across all those databases. So they're models in effect, but I see that, I don't think that's what I'd call a foundational model. So I didn't really answer the question exactly, but you're welcome to ask further. Okay.
I can maybe give a few just thoughts of clarification, even though I'm not supposed to answer these questions. I mean, there's some potential in a way, like you imagine that like I give you some images, then I give you some text. There'll be something in those images related to the text, then I can keep track of it. If I give you some genomics, maybe there's something else that's not mentioned in the text that people don't care about. Maybe I can keep track of it. Now, once I produce like something that represents this image, it keeps track of things that are signal as defined by other modalities I'm collecting that presumably is useful for a downstream task by which I would predict if the label or whatever task I have is also congruent with those modalities. And so there is a potential for it, but like the worry maybe comes in terms of like biases and noise. Like if you have like hey, this is how we get the data because of, I also had a limited understanding of this presentation. If this is how we get the data, we have like certain molecular processes that we're doing. They're leaving a signature inside of there, and we can see what that signature, and even if you give it to me at scale, but because this is how we collect the data, we're going to see that, then you need to have an understanding of what that data is to back out of it.
Just on that, what I am seeing people do, actually I can think of a specific example. In the academic medical center databases, we have say in ours is 6 million patients, but we have images and we have the electronic health record and we have other things. And in some cases, we have sensor data and that kind of thing. So we learn something across those modalities. Then we have these large claims databases that are just the claims data, but there's 100 million people, so they have value beyond the academic. So then we learn how to like extract information from the larger one and bring it down, or extract models from the smaller one and apply it to the bigger one. For example, you're creating a phenotype. So based on the raw data, you want to know if someone has taken a drug or has a disease. If you do it in the large claims database, you'll only have limited accuracy of your phenotype. So then you go to the academic medical center and improve the phenotype because you have the image data that you can corroborate on and learn more about it, and then do the best job you possibly can with what is available in the claims database. Then you go back to the claims database and actually run the study because that has enough patients to find the rare side effects. So we can flip between these database sizes where some are smaller but deeper and some are more superficial but huge.
We have a question.
Second talk, thank you for a very nice talk. This problem of bias correction, of course, arises in lots of different cases, not just in genetic data. And it almost seemed like in this case, there's like a happy coincidence that the biochemistry is sort of generating both the bias and the truth, and you can essentially use the same model. I work more with imaging data where, maybe this is not so much the case. So I'm wondering if you've thought of sort of whether there's a way to do this more generally, or is it sort of, like, for example, you could imagine still learning a neural network for the bias and then having a different architecture for learning the residual or something like this. Are you exploring that at all?
Yeah, so we have some situations where it's not as easy because we don't have a convenient background to learn from. And one strategy that has worked pretty well, but I think it only works in our case because in many ways the simple feature attribution methods and stuff give us really beautiful interpretable features, which is much harder to do with more complex images and things like that. But since there's a lot of known knowledge about causal drivers of biological events, we can in fact just bootstrap the model. So one idea we've tried is, imagine you didn't have the background, you just fitted a model that had the confounding bias. But using the interpretation techniques, you can actually identify pieces of the attributions that are noise versus not, and then just sort of self-loop and eventually you can deconvolve one from the other. I don't know how feasible that is in the imaging case or other cases, but it is an interesting question. I don't know, I mean, I'm sure you know more about this, but I don't think there's an easy solution to it. It would have to be customized for specific types of inputs. It does make me feel more apprehensive in general about, these general learning models which are trying to learn everything. I find it hard to understand precisely how, at that scale, you would be able to pull out the signal from the spurious noise and the confounding. So I think that's a general question I guess everyone's grappling with. Thankfully, in our case, we understand the inputs a lot more, and we can perturb them, so we can figure out causality quite conveniently, although it's a little laborious, but you can eventually get there.
Imaging's not my area, but that's a fun example, because I would think some of the noise is going to match physiology, and some of the noise is going to be like someone left their pacemaker showed up, or a label, or their necklace showed up. So you can have one model that's going to be very different and kind of symbolic even maybe, and the other one is going to, so you're going to need both the physiologic noise and that weird noise that just does odd things in images. That's a fun area.
Actually, yeah, there is one other option which I just realized we have discussed before, is I think if you have an experimental design where you have multiple instruments, for example, assays, each of which will have their own biases, measuring the exact same thing, then you have the opportunity to potentially, again, pull. And I think that works very well for images in the wild. There's so much diversity from the generation process that it's pretty easy to remove the spurious signal from the noise from the signal, because it's not perfectly confounded. So maybe that's an option. So having some diversity in the input generation process might actually help.
All right, I was told I actually have to cut us off, but let's thank our panelists again.